Hope they can hear me. I don't even know if the microphone works. Probably should check on that. Are you able to hear me? Yes, no? Feedback, please. Microphone. Ah, okay, they can hear me. Cool. All right. Questions? And, I don't know what the hell. Whatever. Here to answer questions. Any questions you have, including but not limited to things you should worry about. Aerobic respiration. You know the two slides that say aerobic respiration and oxidative phosphorylation? Wrap your head around those two. Don't worry about the glycolysis explanation and anything beyond what's on those two, really. You also have to compare what happens in the cytoplasm to what happens in the mitochondria as far as what's present and what processes occur, like making ATP or what gases are used. Hint, hint. If you have more specific questions besides like the whole topic of aerobic respiration, what do we need to know how to draw? Ooh, how to answer that one. Well, I think there's one that's obvious because it's kind of a big deal. And then the other one, you don't need to know. I'll do this. You don't need to know glucose and ribose, either glucose or ribose, either of the glucoses or ribose. As far as water, properties of water is important. Remember hydrogen bonds. You should know how they are formed. You should also understand the importance of properties of water and with a highlight on solvency. And what else is there? Different types of carbs, disaccharides, polysaccharides, monosaccharides. Please explain DNA sequencing once more. DNA sequencing. So they take a strand of DNA that they want to sequence, and they separate the two strands. They add it to a test tube actually of nucleotides, free nucleotides, and some of them are tagged, they're dideoxyribonucleotides. And when replication occurs, when it gets to those, they stop. And so now they have different lengths of segments, and they have them separated by lengths through a process that we'll get to later. And based on the lengths and which ones are tagged, they can determine the sequence. Should look over that though. Is the CO2 that is released in the beginning of the link reaction within pyruvate to begin with. The CO2 link reaction. Pyruvate, when we talk about it, I just talk about the three carbons, but there's also oxygens and hydrogens and things like that on it. So the CO2 that breaks off from it is a combination of those. It's not like a CO2 stuck on there. It's just part of the molecule that breaks off. What do we need to know about chemiosmosis? Yes. DDO, dideoxyribonucleotides. Where did that CO2 come from? Goes from 3 to 2. So one of the carbons breaks off, and on that carbon there's also going to be oxygens, hydrogens, and things like that. But when the carbon breaks off, the oxygens go with it. It just kind of rearranges itself. There's enzymes involved, and enzymes rearrange the electrons and things, and from that, the CO2 comes off. What's the answer to chemiosmosis? Yes. You need to know all of that. Those two slides about aerobic respiration, oxidative phosphorylation, they are worth the time. As is DNA structure, DNA replication, course. See what else is there I could give you hints about. The unsaturated fatty acids, cis and trans, the health ramifications of them. Condensation and hydrolysis reactions, how those occur with the hydroxyls and hydrogens and forming water and breaking water. I think what else there is that I could help you with. Do we have to study? No, there's nothing about people or any experiments. You're welcome. That page gets ripped out of your notes right away. Keep it. You're going to need it later. But the only questions they ever ask about people or experiments are multiple choice ones. If I write chemiosmosis, yes, I get full credit. Absolutely. Kinsley, and you can explain the stuff about oxidative phosphorylation. Time for me to explain oxidative phosphorylation without pictures, because otherwise it's just a bunch of words and mumble jumble. So look over the pictures. Here's what I advise you to do. Look over it. If you can download the PowerPoints, then you can kind of go through the animations or the pictures even, if that's all you have. As you read through the explanation, look through the picture, and then you'll see these things happening as you read along. So it'll kind of make more sense. It'll fit in your head like a movie instead of a bunch of words. Do we need to know about the ins and outs of LDLs? Yeah, you should know the risk ramifications with regards to fats. At the end of aerobic respiration, we get 36 ATP. Is that correct? 36 to 38. I think the 36 is where they kind of favor. Whatever one is written, I think 36 is the one that I've written in there. There's 38 if you do the math, but they say that perhaps 2 are used to move the pyruvate into the mitochondria for the link reaction. So those range. Trying to speak more clearly. I don't know if I'm succeeding. Question for you guys, because I'm not sure. Are people able to see this later if they're unable to make it to this little gathering? Can they watch it later? Just a yes or no is fine. What is acetyl-CoA used for? The acetyl is the 2 carbons that are attached to oxalacetate. Thanks, Nick. The CoA is just so that it can attach. It's like a transporter. It brings it there. The 2 carbons attached, then the coenzyme A goes back and gets another one, and it keeps repeating. So the acetyl-CoA attaches the acetyl, which is 2 carbons, to the oxalacetate for the Krebs cycle. The acetyl is a 2-carbon molecule that's attached to it. So it's acetyl and then coenzyme A. Oxalacetate is the 4-carbon molecule that the 2 carbons attached to. Those 4 equals 6. 6 minus 1 from a carbon dioxide being lost is 5. Another carbon dioxide is lost, it's 4. Just back to where you started. Could you explain how respiration is measured? I can, but you needn't worry about it right now. But I can explain to you. So in the picture that we had, and again, it's hard to do without an image. On one side, there are seeds going through respiration. So they take in oxygen, give off CO2. And on the other side, it was to equal the volume so that the water was balanced. So when the seed, but on the side with the seeds, there's a solution in the bottom, potassium hydroxide, that absorbs CO2. So when the oxygen is taken out and the CO2 is given off, instead of going in and equal in volume, it gets dissolved. So now the volume decreases, and it causes the water to suction and pull towards it. You can measure how far it goes. Is the Krebs cycle I don't know what that means. Do we have to compare lipid and carbohydrates? You mean the energy storage? Because otherwise I don't know how you would compare them. The things that are made during the citric cycle. For our purposes, no. The extent to which you need to know about the link reaction, glycolysis, and Krebs cycle is in those two slides with the aerobic respiration and oxidative phosphorylation. Are there any specific molecule structures we should be familiar with? I already answered that, but you don't need to worry about either glucose or ribose, like the properties of each. I don't think so. Wait, which process are the that's the Krebs cycle. But as I said, the extent to which you need to worry about that for right now is what's in those two slides. Will we have to know the difference between methane and water? No. Links to the, yeah. James is helping me out here. Because it's a cycle. It starts over and over again. Could you please explain the significance of telomeres? Ooh. Telomeres are the extensions from DNA. I don't say extensions. The parts of DNA that are after the genes that actually code for something. Because every time DNA replicates, it doesn't go all the way to the end. So a piece of it in the next generation of DNA that's been replicated is shorter than the previous one. So every time it's made, it gets shorter and shorter and shorter. And the telomeres are what allow it to do that without getting to the genes and interfering with anything else. So they are important for that. There's also introns and genes for tRNA and for gene expression, the promoter. You might want to look over that. Introns, we will get to that next section. It'll make more sense then. I missed something. Can you explain what happened to Maslow's self experiment? I can, but you don't need to worry about it. They were trying to determine the proper what's the word I'm looking for? Model for DNA replication. And they did the experiments with the N15 DNA, and the free nucleotides that are being made to copy DNA were N14. And by taking the masses, they were able to determine that it was semi-conservative. But again, you don't need to worry about names and experiments on this. Can you explain the condensation reaction? I don't understand how water joins the two. Water is made during a condensation reaction. So an OH from one and an H from another break off and they form a water, H2O. And then now the two molecules have a bond because one was attached to an OH and the other one was attached to an H. So now they bind to each other. Introns, we'll get to introns next. Just know that they exist for right now. Introns, they're cut out of mRNA after transcription and they stay in the nucleus. And we'll get into that more. Just right now, know that it's part of the DNA that doesn't code for anything. Genotype of starch, correct? Know the saccharides in general, mono, di, poly and stuff. Yeah, just know which ones are which. Mitochondria, does the link reaction occur in the matrix? I think the link reaction is in the mitochondria. It is in the matrix. So again, areas of focus: DNA structure, replication, aerobic respiration, the aerobic respiration and oxidative phosphorylation ones, water properties, hydrogen bonds, specifically solvency, health factors with lipids. You're gonna punch this test in the face? I don't know what that means. Think the link reaction. To what extent does you don't have to worry about that mean? Like, don't have to worry about it at all, or for the test? I mean, it's if anything that's on there is eligible to be on the IB exam in May. I'm just telling you the stuff that you. I don't want you guys up till 4 o'clock in the morning with a bunch of stuff and then get pissed off because it wasn't on the test. So I'm trying to help you funnel it down. Usual fatty acids doesn't matter how many carbons we put. No. But why? I don't understand the question. This is like the best part of biology. I get it, but also I can see both sides of it. Matrix, I knew that. I mean, good job. What happens in the cytoplasm of mitochondria with respiration? Different types of unsaturated fatty acids. The drawings I told you about. Condensation reactions, hydrolysis. Ooh, BMI. There's a graph question for BMI. So you don't have to necessarily do any math. You just have to be able to read a graph. Different carbohydrates, disaccharides, monosaccharides, water, molecules, got that. Carbs and lipids. This is cis and trans, unsaturated fatty acids, health ramifications. I think I already talked about that. When you draw fatty acids, are we allowed to write COH on the right side like an amino acid? Yes. When you draw DNA, how do we know which one is the 5' end? The 5' end is the one that has the phosphate, and the 3' end is the side that does not. There we go. What else? DNA structure, replication, DNA replication, the enzymes and stuff. Rapid respiration, that. Yeah, I think I pretty much summed it all up as far as the focus. Do we need to study redox stuff? That's just for vocabulary purposes. So you knew what oxidation meant and things like that. And that they were, when we get to photosynthesis, you'll see it's reverse. So that was the whole purpose of that. And for vocabulary, you label the 5' end. That is up to you. What are these things called that hold the separate strands apart? Those are single-stranded binding proteins. They're like pins. Imagine like pins, like holding. Remember when you were in middle school and you dissected a frog or something and you held the skin apart and you put the pins in? It's kind of like that. If you didn't dissect a frog, then it's like pins holding two things apart. Again, you don't need to worry about people in experiments. You don't need to worry about the glucose and ribose. DNA sequencing wouldn't be a waste of time. The reason why I waited until now to do it is because I want you to study some stuff that's not going to be on the test because you're going to be responsible for it later, but I don't want you hurting yourself staying up all night. So I like to think you've started studying before now. So some of the stuff I'm saying you don't need to worry about, it's not like you're not going to study it at all. You already did, I hope. But just, I don't want you guys being up all night. Do we have to know a lot about proteomes? We haven't done proteome. Jack, we haven't done that yet. We haven't done anything about proteins or proteomes or protein synthesis. Just look at the presentations that are in the first file. Don't look at all of them. If you printed out the review one, then you're going to be really confused because it's all mixed together. Will there be labeling? Anything you draw, you should label, especially big things. think I printed the wrong one. Yeah, it's very possible. uh, is there anything else? I think if you listen to this back, and I don't want to make it too long because then people may not sit through it again. If you just listen to the things I told you to concentrate on, then it should at least funnel the studying that I hope you've already begun. So it's not like the other stuff isn't being learned at all, but maybe not. Like I said, I don't want you being up all night. We have to know about fermentation. That it's actually, I don't know if you need to know anything about fermentation. But for fun, in animals, it makes lactate, and in plants and yeast, the pyruvate becomes ethanol and CO2 in order to get the NAD to regenerate for glycolysis over DNA sequencing. To what level of detail do we have to memorize respiration, all of those cycles? If you look at the PowerPoint on aerobic respiration, and then the following one on that continues aerobic respiration with oxidative phosphorylation, it's numbered. That should be sufficient. DNA sequencing, again, is they take the strand of DNA they want to sequence, and they put it in a test tube with nucleotides. Some of them are tagged. They're called dideoxyribonucleotides. And when they attach, DNA replication stops. Then they separate the strands by size, and small to large, they can tell which nucleotide is present based on the length, and then they can find the complement, which is the sequence that they're trying to figure out. Please don't tap me, but what is chemiosmosis? Will we have to draw the, no. I never said you had to draw the electron transport chain. What is chemiosmosis? Chemiosmosis is the proton gradient that's formed from the electron transport chain and the diffusion of the protons through ATP synthase to make ATP. On the tests lately, the big tests in May, they've been a wee bit more specific on things. So details are best if possible. I got your back. You guys have 6 classes, and this is but one of them. And for some reason, you guys end up having multiple tests on the same day. Do you have to draw any reactions? No. The only things you have to draw are 2 molecules. One I think you can figure out, and the other I've hinted too much. You don't need to worry about the glucoses or the ribose. So you got the amino acid, the fatty acid, and DNA. 2 of them will be on there. You're welcome. Any other questions or anything? You think that's it? Anyone else have anything? Where's Anna? She's in her room looking over her notes. Thank you, Sandy. Yeah. If there's nothing else, then I'm going to end this at 25 minutes. So 20 more seconds of anything you need. You're welcome. You're all welcome. Proud. The triglyceride is what exactly? Triglyceride is a glycerol with 3 fatty acids. It's a fat, which is a type of lipid. You're welcome. You don't have to keep saying thank you. It's fine. 3 lipids. Okay, good. Okay. All right, 25 minutes. See you tomorrow. Good luck.